recording now. So take it away, Allison, to introduce our speaker.
Go team. Well, it gives me great pleasure to introduce Mikaela Hintz, who's been so much fun to work with. Mikaela came to me earlier in the fall term, fall term, it all runs together, with this wonderful question of, do you understand how basically quantum biology, what are your thoughts? And I'm like, I don't know. And so she brought these wonderful papers and we struggled together to read and understand some of them. And just this, it was so wonderful to watch her develop as an innately curious student, to tackle these challenging problems and understanding this glorious realm of biology, with quantum mechanics smeared throughout. So I had so much fun learning so much from her, and find myself being more and more inspired to learn biology now. So thank you, Mikaela. Anyway, so I'm very excited for her to share the work that she's showing today. And then, yeah, hopefully soon we will see news, science articles and such, of the quantum biologist that she will become. Take it away, Mikaela.
Thank you, Allison. That was a wonderful introduction. So like I said, for about the past 6 months, I've been exploring the topic of quantum coherence in photosynthetic systems. But before I get into the real science part of my talk today, I want to share a little bit about how I actually arrived at this topic, because I think it's important to talk about what goes on behind the scenes of science, and to remember that science is a very human-driven process, and it's so much more than facts presenting themselves to us. So I came to Lawrence as a biology major, and that was fine for the first year, but like midway through the summer after my freshman year, I was working in a biology lab on campus and was quickly coming to realize that I didn't think the field of biology was where I was going to find the work that I felt really passionate about. And I began thinking that maybe I should consider chemistry, and that it might be a lot more interesting than what high school had made it out to be. It didn't help that all the chemistry kids on the second floor just seemed to be having lots of fun. I got to play with the fancy machines in the basement and mix cool chemicals. So at this point, I wasn't really sure, and if we're being honest, I had like no idea what I was interested in, but I had it in my head that like science was all about answering questions, and like any bored and overeager undergraduate student, I wanted to find the most compelling one I could and let that kind of guide the rest of my academic life. This led to the following Google search, probably about mid-July 2017, biggest unanswered questions in science, and that's pretty much word for word the search I typed into Google one Saturday in the Coleman loft. I really wanted to know what we didn't know, and the results led to me to a page with a list of like unexplained fields and questions, including quantum biology. And that really piqued my interest and held some potential, but kind of got put on a shelf once the school year started. The beginning of winter term sophomore year, I became a chemistry major and had the opportunity to spend the summer making a whole bunch of chloroxylene molecules with Stefan for his anti-schistosomiasis project. And even though I was kind of exiled to the teaching lab by myself, it was a whole lot of fun. And that experience had me pretty convinced I wanted to go into synthesis. But when it came to my senior year, my interest had shifted, and I kind of found my way back to quantum biology. But this time I was armed with much greater skill set to engage with the research out there. And also I had the guidance of Allison, which was invaluable. Could not have done it without her. So quantum biology. The field of quantum biology, it looks at biological models and processes on kind of like the molecular scale, and then asks whether we need to take into account quantum mechanical concepts to explain what we see going on. This is applied to a long list of things, including, but not limited to, our sense of smell, and why we can differentiate between the sense of very similar chemical compounds, as well as magnetoreception in birds to explain how they navigate while migrating, the human consciousness, as well as enzyme action, and most importantly for us today, photosynthesis. So my preliminary research into the literature revealed that there was a large body of work into how this theory could be applied to the photosynthetic process. In particular, a large field seeks to address the questions of why photosynthetic energy transfer is so incredibly efficient. And before going any further, I want to make it clear that when I say photosynthesis is very efficient, I'm talking about the energy transfer between structures within a photosynthetic system, and not the efficiency of light energy being converted to chemical energy in the form of glucose sugar. So in order to understand this question, we need to understand our system and what's going on in photosynthesis. Because the literature I read utilized photosynthetic bacteria, in particular green sulfur bacteria, instead of plant-based molecules, that is the specific process I will be going through. Though it is very similar and translatable to plant photosynthesis, we have like an overall process that can be broken down simply as the absorption of light energy driving the light-dependent reactions that produce carbohydrates and oxygen from carbon dioxide and water. So initially in this process, what we have is the light energy is captured by pigment molecules after passing through what is known as the antenna region of the photosystem. And it creates this temporary excited electronic state of the pigment. This slide shows a specific complex called the Fenner-Matthews-Olsen complex. This complex is derived from green sulfur bacteria and is used extensively in lab research because of how well characterized it has become. Many of the papers I read use this isolated complex, and if you look closely, you can see that it consists of a trimer of 8 bacterial chlorophyll pigments bound to proteins by a magnesium middle center. And once excited, the energy delocalizes across all these chlorophylls and is then channeled to the reaction center region of the photosystem. The reaction center is a pigment protein complex and then it is a lipid bilayer. Once the exciton reaches the pigment in the reaction center, then that's where you have your charge separation. That's where your charge separation occurs across the membrane. And this charge separation starts off a chain of electron transfer reactions that creates the proton gradient across the membrane that powers processes like the synthesis of ATP. Because this energy movement through the chlorophylls is so important, I'm going to zoom in a bit closer and walk through the process here. Here we can see or get a better look at how the chlorophyll rings are spatially coordinated in relation to each other. Since we've peeled back the protein, there we go. Sorry, it's a bit laggy. You peel back the protein. There we go. Okay. So we've peeled back these protein sheets, and we can see that as the exciton makes its way down to the reaction center, there's a decrease in energy with each of those steps down. And each of these 8 chlorophylls actually has a different excited state energy. And the exciton will be transferred from highest to lowest down to the reaction center. The arrows here are a bit confusing because they don't correlate to energy movement, but they're there to help indicate like the positioning of the pigment in space. So as we're thinking about energy transfer, we're saying that the initial excitation of the pigment molecule results in the creation of an exciton. And exciton is created when an electron of a molecule is excited to a higher energy orbital from its ground state. Now, not only do we have an excited state electron, we also are left with this hole that it once occupied. And we kind of think of this as an electron and a hole pair, and it kind of moves from molecule to molecule in this complex, in our case, like chlorophyll to chlorophyll. And the stronger the interaction there is between those pigments it's moving around, the faster the exciton will move. Those initial energy transfer events that occur within a range of hundreds of femtoseconds to picoseconds, and the quantum efficiency is near unity. As I've mentioned earlier, in order to explain this efficiency, some have suggested that quantum mechanics may provide some useful explanations and potential mechanisms. As new and as exciting as the blending of biology and quantum is, it actually has a pretty decent history already, including work of James Franck and Edward Teller, who in 1938 actually came up with the mechanism for excitation energy transfer photosynthesis that actually did involve quantum coherence. This was more of like a hypothesis at the time, though. I keep using the term quantum coherence a couple times, so this is a good place to stop and define what that means. Electromagnetic waves can be described by a characteristic called coherence. Essentially, this means that the waves are like in step or in line with each other. This feature isn't always like described as a binary characteristic, but more of like a spectrum, where radiation can be assigned a degree of coherence. For example, lasers like this down at the bottom are described as very coherent, as the waves they emit line up for a very long time before becoming so misaligned that any semblance of their like coherent characteristic is lost. At the top, you could think of this as radiation from the sun, which produces very incoherent light and is made up of a wide spectrum of colors and wavelengths. And something like a single colored LED is a good example of light that's incoherent, where its wavelengths don't line up, but it's all the same color, all the same wavelength. They don't happen to line up coherently. So all of these light sources can be assigned both the coherence length and time, as we're thinking about the degree of their coherence, and a general relationship between bandwidth or the range of frequencies of light to the magnitude of the reciprocal of the coherence time. So this isn't an exact equal sign. It's just that these are in the same magnitude as each other. So if we do a little bit of rearranging here, we can pretty easily see that this also means that greater bandwidths or greater range of frequencies present results in a shorter coherence time. And now we can look at the relationship between coherence length and coherence time, substitute in our relationship coherence time and bandwidth, and now we can see that the greater the difference in wavelengths, the shorter the coherence length and vice versa. A very coherent laser can have a coherence length of around 15 times 10 to the 6th meters long, while light from like a white light bulb has only around like a 900 nanometer coherence length. There's a huge range there. The presence, nature, and like the significance of electronic coherences presented by excited photosynthetic complexes has been the center of debate among researchers since all the way back in the 90s, when several papers came out claiming to have witnessed spectroscopic evidence of coherence in bacterial photosynthetic reaction centers. The presence of coherence was a topic of debate for several years, but with improved spectroscopy, it has become widely accepted that there is hard evidence of coherence within both bacterial and plant complexes. Once it was established that there was some non-trivial coherent signal, the next debate became about the determination of such signals as long-lived that I found in many papers that I was reading. And it feels uncomfortably subjective. So one important way of defining what long-lived means in this context is to look at the scale of the energy transfer occurring within the same complex. If an argument is being made that the coherence is relevant and important for efficient energy transfer, it has to survive for a similar time scale as the energy transfer. If coherence is breaking down long before the excitonic completes its journey, then it's unlikely to be helping out energy transfer at all. And this is where disagreements still exist about photosynthetic quantum coherence. For example, these two papers, which will come up in a second, came to fairly contradictory conclusions, despite both calculating coherence time to be around the same 60 femtoseconds. So it's not so much that they disagree on the fact of the coherence happening, but on its significance. So this one paper, so the nature does not rely on long-lived electronic quantum coherence for energy transfer. And on the other hand, we have a communication that says coherences are observed in vivo and that they are used for energy transfer. They make a very firm statement in the conclusion that there is no long range, the paper on the left, that there's no long range coherency, and that it's not an important factor to consider when we're looking at energy transfer efficiency. And however, the paper on the right makes an opposite claim that coherence lasting around 60 femtoseconds is perfectly long enough to warrant further investigation into its potential effect on energy transfer. So once we had kind of established the existence of what we're calling long-lived coherences, there still remained this question of what was going on within the complexes that allowed coherences to exist for long without dephasing, especially in such like a biological noisy and wet environment. We know that coherences exist, but we don't know why yet exactly. And we haven't discussed how these researchers obtain their information in this talk yet and what kind of techniques they use to obtain information about the quantum dynamics of these pigment protein complexes when they're so complex and messy. So this is where two-dimensional electronic spectroscopy comes in. 2D is essential spectral complexes. It is the main experimental method used by any group exploring photosynthetic quantum characteristics, and some literature I've read on it refers to it as the optical analog of NMR. 2D ES utilizes multiple lasers in a pump probe setup to develop plots that relate excitation and then emission energies as a function of the waiting time between excitation and emission events. So within that frequency of the laser pulse is where that's happening. So it's important to know what kind of laser is being used. 2D spectra are then plotted from this data as a function of absorption and emission. The mechanism of 2D electronic spectroscopy can vary slightly from experiment to experiment, but in general, the process begins with this initial laser pulse that strikes the sample and then causes an exciton to become coherent with the ground states. These coherences propagate out and can be described by the equation right here under coherence. H sub G representing the ground states and the H sub EI representing the exciton. The inherent difference between those two H terms describes the ground state wave and the exciton wave properties. The differences between the two can cause the system to evolve phase at a frequency that's described right below. Meanwhile, the nuclear motions of the system perturb the excited state transitions, making that frequency change over time. So if any of the individual complexes experience a disturbance that the rest do not, the complex will deep excited state energy gap. That will show up in electronic spectra when we're looking at our 2DS spectra. After this initial first pulse and the allowed coherence time has passed, a second and third pulse will occur that reverse the phase evolution the system has undergone with the first pulse, but then allow it to re-evolve in the rephasing time. So what we have here is a nice graphic for visualizing the timeline of a typical a 2D electronic spectroscopy experiment. However, this isn't what a 2D electronic spectrum looks like. This is just a convenient visual of the timeline of that experiment. On this next slide are some examples of simplified 2D spectra. These aren't coming from any experiment. These were just created to demonstrate some of the information that you can gather from the spectra. One of the most important characteristics is to look at peak position. The peaks that are found along the diagonal line are appropriately called diagonal peaks. They appear when the system emits a signal that's the same frequency as the signal that excited it in the first place, and also reflects information that a linear absorption spectra would give you. These peaks off to the side of the diagonal, those are referred to as cross peaks, and those are important later. These appear when the system would emit a signal that oscillates at a different frequency than the excitation signal. So when a cross peak appears, that means that there are two electronic states of the system are coupled, because when the pulses create an electronic state, the system responds with emission from a different energy level. And this coupling can be related to energy transfer or charge transfer between molecules. The other characteristic over here on the right that can be observed is just the general shape of the peak. You can see that here on the right, there's a very distinct roundness of the peaks. And the top one is very oblong. The more oblong the peak, the shorter the population time. The width along the diagonal line at the top represents the inhomogeneous broadening, which would contain your information about interactions between the environment and the system. So your complex system and then the proteins that are wrapped around it. If the system is composed of like a large amount of identical molecules, each of them interacts with the protein or the environment in a different way, kind of exactly like our chlorophyll rings in the FMO complex and other protein pigment complexes. This would imply that the same electronic state of each molecule assumes a different but small variation, and the line width along the up diagonal kind of shows a smaller value with respect to the diagonal one. In this case, the spectral broadening contains a contribution from what we call local interactions of the system, and that would indicate that the width reflects homogeneous broadening. Okay, now that we have some of the basics of spectroscopy, we can go back and readdress the question we posed earlier. That is, why in the world do we see this coherence in the first place? And that question was actually recently tackled by the Engel group from Chicago in a paper that made a surprising discovery regarding the protein environment of the electronic coherence. Their motivation for the study was kind of the fact that the typical means of slowing down dephasing from coherence were impossible for a biological system to really take advantage of, like lowering temperature or physically separating the coherent states from their environment. That's not something a cell or protein can accomplish. So with that in mind, they really needed to figure out a feasible explanation for why these complexes continued to demonstrate coherences at kind of physiological temperatures, not cryogenic temperatures that we see in labs. So in order to determine how the nuclear environment and the exciton affects its movements, the group decided to develop two different spectroscopy methods, which is pretty impressive. The first is called two-dimensional single-time domain exciton perturbation spectroscopy, or shortened 2D STEPS, which is much easier. This type of experiment measures how the excited state nuclear motions systematically perturb individual excitonic transitions after excitation has occurred. So after that first pulse excitation by the laser, 2D STEPS measures the dependence of the frequency during the coherence time on the coherence time in order to find out what kind of vibrational dynamics are going on in there. And then the second experiment that Engel developed is called 2D trips. And instead of looking at how individual excitons are affected by like a systematic motion, 2D trips looks at how one exciton's motion can affect another unoccupied excitonic state. So the spectra for trips is significantly different from steps, in part due to the fact that trips looks at both the coherence time domain and the rephasing time domain. So during a TRIPS experiment, the nuclear motions of one exciton is observed like in the coherence time domain, and the spectral motions of another exciton are observed in the rephasing domain. So as the coherence time for the initial exciton plays out, the effect it has on the exciton motion in the rephasing time is observed. So any dependencies that are seen between the two excitons, that would assume that there is some correlation between their nuclear environments. So after developing these spectroscopic techniques, the group went on to perform 2D steps on 7 of those 8 bacterial chlorophylls that are in the FMO complex and their respective excitons. They did not perform this on the 8th because it's has significantly weaker bond than the rest, and functionally it just mostly serves to like hold the whole complex to the chlorosome base plate. So that one was left out. This Nile blue that you see here in the right hand corner was used as a model system verification that the experiment worked correctly and was providing accurate information. So these full color gradient 2D spectra can be like super overwhelming to look at first. There's so much information going on. So what they did to better compare like the motions of the 7 excitons is that they isolated that center spectral density line in one chart down here at the bottom. So with them all lined up like this, it's super easy to see that they had some coherent quality with all the peaks kind of neatly lining up together. And then this indicates that even though the 7 excitons all originate from different site compositions, because of the different pigment protein bonding, you still see very similar vibrational motions, even over the duration of a picosecond, which doesn't sound like much, but it's a lot in this context. So after running 2D steps, the 2D TRIPS experiment focused on the effect that an exciton's motion in the coherence time could have on another exciton's motion in the rephasing time. This occurs over such a small time domain range that the group had to focus on a comparison between excitons 1 and 7 in order to avoid energy overlap that would make like a spectra impossible to read and like accurately assign peaks to. In this spectrum, we see at the top spectra that represents different time points within the coherence time, and then these semi-transparent lines across the heat map show us where those representative slices would contribute to the map information. So this heat map gives us a relationship between one exciton's motion in the coherence domain on a later exciton in the rephasing domain. This black peak line across the heat map here demonstrates that changing rephasing time position as a function of the coherence time position. And then this dashed line here is just to give kind of a reference point. That's a line with the slope of 1. And basically the take-home message here is that if there had been no correlation between those two excitons, if they hadn't been affecting each other, that line would have been horizontal with no slope. But you can see here it almost perfectly aligns with a slope of 1, and that you're taking that to mean that there's a strong correlation between those two excitons. So this paper concludes that the following excitation of the FMO complex shows synchronized and correlated excitonic nuclear environments. So from the 2D steps results, we see that each exciton shows similar motion after excitation, meaning that within these experimental conditions, there were superpositions of 7 excitons that were coherent for at least 1 picosecond like we saw before after that initial laser pulse. So that's represented here at the top, where we see synchronized nuclear motion after simultaneous excitation. And then the 2D TRIPS experiment showed us that one exciton's spectral motion affects another distinct one. And the paper states that these properties are probably the result of the structure of the FMO complex and its chromophores being situated really closely to that protein scaffold they're embedded in. So we've seen a lot of work on the FMO complex, and it works really well for spectroscopic experiments and comparing data because of how well characterized it is. However, green sulfur bacteria are not the only photosynthetic microbes out there that provide interesting quantum considerations. For instance, we have what is called a cryptophyte algae. They have also been studied, not as extensively as FMO, but pretty well studied, and in the context of quantum biology, with particular interest in the quantum coherence that you can see within their bilin pigments. They have different pigments than our FMO complex. These cryptophytes have 8 pigment molecules within their complex, just like FMOs, but instead of identical bacterial chlorophylls like FMO does, cryptophyte has several different bilin molecules, and because of this, cryptophytes are able to absorb a greater range of light wavelengths. What makes cryptophyte algae of such great interest is that they somehow still produce quantum coherence within their photosynthetic complexes, despite that huge and unusually large gap between their chromophores, which is around twice that is found in plants. Closely packed chromophores, but not too close to overlap, are what you're typically expected to find in order to maintain or even like ephemerally produce some kind of coherent effect. And it would certainly seem required for any kind of coherence that would be called long-lived. But the cryptophyte algae doesn't really seem to care though, because it's been researched and found to produce a coherence time of 130 femtoseconds, which is over twice as long as the FMO and other complexes. The authors of one paper regarding coherence in cryptophyte algae theorized that this correlated protein motions, like we saw in the FMO before, might be slowing down decoherence and facilitating this longer coherence times. And then they go on to note that in cryptophyte algae, unlike most of the photosynthetic pigments, in cryptophytes you see their pigments are covalently bonded to their protein scaffolds, while other photosynthetic complexes are not. So that kind of brings me to an analogy that Allison once told me about this coherence and like correlated motion. It's like as if you're on the swing and you grab the other person on the swing, and the stronger the bond between you two, the more the other person is going to kind of take on your motion. That's kind of a fun way to think about, maybe that's the reason why there's a greater coherence in these cryptophyte algae, because they have a much stronger bond between their proteins and their pigment complexes. So this is all really fun to think about, but what exactly practically can we do with this quantum biology? Especially one of the most obvious and well-studied things is the potential for applications for solar energy, especially synthetic solar cells. Our current solar cells aren't that great efficient-wise, and as we've seen today, nature is super good at being efficient about transferring energy. So that's kind of the main focus of any kind of practical applications of this work. Right now, there's not a lot of concrete knowledge about these complexes in terms of their quantum coherence. So a lot of the work being done right now is just kind of to know instead of to come up with really practical applications for it. But in the future, I think in the field of quantum biology, I would hope we would see more concrete answers. A lot of times in these papers, you see these spectra and they have their interpretation of it, and it's not super concrete. They say things like, we believe that this may suggest that this is probably vibrational and it just uses a lot of unsure language, which isn't really their fault. We just don't have the technology or knowledge to know that stuff yet. So I hope in the future there's more advancement in spectroscopy. There's already been some really cool stuff that we've seen today with that invention of those 2 new spectroscopy techniques. And hopefully, as quantum biology moves forward, there's also more practical applications, because I'm not against knowledge for knowledge, because I like that a lot, but also it would be great if science also fades to sometimes some general takeaways I had from kind of doing a literature review is that sometimes good science is not presented well, and you have to slog through bad papers to find the answers you want. And also, as Janice taught us, the papers that were the most persuading and best to read were because they were really well organized and they were written with good prose and really clear graphics. And those were so much fun to read and so much more persuading. I wanted to believe what they were telling me so much more when they made it easy to read. And also, kind of a fun thing I found is that because of the conflicts of opinions about stuff, it often came across in these scientific papers. And sometimes groups were not afraid to call out other groups about how they thought they were really wrong, really wrong about things. So I guess to wrap up, I just want to say thank you to Allison. I basically just walked into her office, as she said, and she totally sat me down and said, I don't know anything about this, but we can learn it together. I also want to say thank you to Deanna, who puts way too much time and effort into us, even though we don't deserve it sometimes. Also to Stefan, who got me into being a chemistry major, and then all my friends and family who helped me out. I'll take any questions now.
Yay!
I would like to thank Mikaela for persevering through some of these technical difficulties. I'm so sorry we're having the technical difficulties, Mikaela. You handled it with grace, and I just want to compliment you on that. With that, as Mikaela said, she'll take any questions. Did anybody have any questions? Just shout out. I'm getting worse and worse as Zoom as time goes on. No questions?
So you mentioned smell as one of the things that this might be part of. What do you think about that? Is that actually legit, or do you think that's maybe going beyond what we actually know?
At first it sounded super fake, but then when I read about it, it actually made a lot of sense. They talk about how our classical kind of view of a lock and key model for that kind of process doesn't explain why there's a difference in, if you degrade a molecule, it smells different, and that lock and key model isn't fine enough to tell the difference between those two. But looking at vibrational characteristics would explain that. So it makes sense from the way it was presented to me.
Okay.
I have a question.
Go ahead,
Oh,
Allison. Go.
no, I was just gonna ask. So as we learn that some of these processes are possible in some of these more complicated systems, is there any chatter in the literature about applying this to some of our constructed, semiconductor materials, or some of the more complex, semi kind of nanostructure type, maybe liquid crystal-like structures?
I've seen things about building better photovoltaic cells,
Mm-hmm.
but I I don't know that I've seen. I think it's just because my focus was not in kind of the material side.
Yeah.
I'm sure there is.
You're just
that
with
drive
the
to apply it
grandeur.
that way.
The literature searches you did at the beginning and like in the introduction, they normally say, you know, oh, it's possible application, or in the conclusions. It's just as you learn more and more about some of these complicated pathways, it seems that, I don't know, there might be information to tap in a completely different field, or to transfer to a different field.
Well, and to jump off of
Yeah.
Allison's question and to kind of bring it back to where you started, you know, there's two ways to kind of conceive of big unanswered questions, right? There's the kind of curiosity based, like, how does this work? And then there's the sort of like big societal-ish like problems we need to go solve actively. And do you see this kind of playing any part in the latter? Or do you see this as more of just, you know, breaking down very basic science, figuring out how the world works?
I think at first it was just about, we want to know what's going on for the sake of knowing what's going on. But I think that it's moving a lot more towards fixing our energy problems and coming up with greener energy sources. That's kind of the draw. I don't know if that's just because they need to think of a reason to fund it, or if they actually are that interested in trying to solve that issue with this method. It would be really cool if it really worked out, because I don't know, but from what I've heard, photo cells right now aren't that great, and nature does it really, really well.
Yeah,
So
increasing.
it'd be great if we could tap into that.
Increasing that quantum efficiency by 5% would just have such a massive impact. Excellent, thank you.
I guess that my question was really, so with these brand new methods in spectroscopy and trying to engage with like a brand new method in spectroscopy, did you have any sense of like what sort of headspace you have to even get in to think about developing a new spectroscopic method? Like, that seems really like that seems like a really big step to me. And how did, any impressions from your work in looking at these odder ones and these newer ones?
So the spectroscopy that they like developed wasn't like, you know, like totally new. It was based off of two-dimensional stuff that's been done before. They just how something affects something else, and so they need to develop a system where they could probe one thing and see how they could like probe another thing. So I can kind of see the pathway, because it's not like they built it from scratch. They just needed to tweak what was out there to get the answers they wanted. I think you just have to think very three-dimensionally to develop, I think, what they did.
Thank you. Any other questions? Okay. Well, let's thank Mikaela for her time, and I would like to thank all of our speakers for the whole term. And feel free to hang out and talk to Mikaela afterwards. Otherwise, I will see you when I see you. I don't have any more talks to promote, so thank you all for joining us for this talk, or for however many talks you did join us for.